Come again to my YouTube channel. Today we'll be talking about immunoglobulin structure. Yeah, so immunoglobulins are a family of glycoproteins, and based on chemical and structural differences, they are classified in isotypes, which are IgGM, IG, IgG, IgA, IgD, and IgE. So every immunoglobulin has two small chain. We have the light chain and the heavy chain. So each IgG molecule is composed of two identical light chains and heavy chains, which form a Y structure. So the upper two arms of if it forms like a Y. So the upper two arms form an antigen recognition function, which is referred to as the antigen binding fragment. So the lower stem of the molecule, it has an effector function and it interacts with other components of the immune system, such as the complement and cell receptors specific for this type of the molecule, which is referred to as the crystallizable fragment. So we also have another fragment of immunoglobulin, the FV. So this one is going to retain the complete antibody binding sites, and it is going to consist of regions of the heavy chain and light chains containing the N terminal half of the fab. So these fragments have been generated from, let's say, the use of a DNA recombinant expression system. And clinically, for example, they are used in treating cancer patients with mouse monoclonal antibodies, mostly specific for the tumor antigens. So these both light and heavy chains, they are further referred to as variable and constant regions. So light chains contain two domain, the variable light and the constant light. So the heavy chains contain one variable, which is the heavy domain, and either three or four constant domains depending on the immunoglobulin isotype. Yeah, so the Y-shaped immunoglobulin molecule is a proline-rich region between the first and the second domains of the heavy chain called the hinge region. So this region contains cysteine residues that allow linkage of the heavy chain polypeptides to each other by SS bonds. So this region confers flexibility of the molecules. So because it provides mobility of the two upper arms of the molecules, so this is going to enhance their antibody binding potential. So two immunoglobulin isotypes, we have the IgM I mean, and IgE. They do not have the hinge regions. However, their first domain and the second domain region perform a hinge-like function. Immunoglobulin isotypes are named by their heavy chains. So we have like maybe alpha, gamma, yeah, and they contain two types of the light chains. Okay, so what are the functions of these immunoglobulins? So sometimes they can be found they can either be found on transmembrane proteins on the surface of the B cells, or they can be secreted by the terminal cell of B cell differentiation, that is the plasma cell. So immunoglobulins, they function as antibodies and have the property to combine with the antigen that triggered their production. So this property of recognition is referred to as specificity and is controlled by assortment of genes which regulate the production of individual parts of the immunoglobulin molecule through determining the primary amino acid sequence of these components. So immunoglobulins are specialized molecules that provide two functions: an antigen recognition function and an infector function, like we said at the beginning of this video. So these functions are very important during immune response. So the antigen recognition property of the immunoglobulin molecule confers its exquisite specificity to react with different molecular structures. We have epitopes which are either linear or morphine conformational configuration, that is an antigen. So, the effector function of an immunoglobulin is related to the specific isotype produced. For example, we have IgM antibodies synthesized early in the immune response. Because of their large molecular size, uh, they are found and best function within the vascular system. So we have immunoglobulins of lower molecular weight, like the IgG antibodies, which are produced later in the immune response and can readily diffuse between the intravascular and interstitial tissues where they function more effectively. That's all for today. So in the next video, we are going to be discussing the function of each independent immunoglobulin. We'll be discussing IgG, IgM, IgE deeply. Thank you.